Good evening, viewers at home. This is the improved audio version of our previous lecture on animal breeding. Just like we have it in the previous video, this is animal breeding and introduction to animal breeding. Animal breeding, what do we mean by animal breeding? Which is a deliberate attempt to select and mating of animals in order to ensure or to enhance desirable traits. So the reason why we improve animal breeding or when we look at breeding in animal is number one, improving productivity. For example, this is an example of this. Then another one is in terms of their health. So diseases resistant to be able to resist different types of disease resistance. Then another one is to be able to for economic reasons. Economic reasons is one of the reasons why we also improve animal breeding. Okay. What are the importance of animal breeding? Just like I said, in terms of a greater meat production, improve meat, egg yield per different animal. So just like I said, and it's also to boost reproductive efficiency so as to be able to reduce the generation interval and be able to produce a higher fertility rate. So then also to promote environmental adaptability, that is a breeding that is suited, that is actually locally made, suited for the climate change in terms of feed resources. Okay, the another one is to be able to drive economic growth, which is as a result of higher profitability and food security. Okay, the historical background, just like I said, that the issue of animal breeding actually started from the early domestication by human of animals. So domestication of animal for food and for to be able to help them in their food in their farms. Okay, and by the 18th to 19th century, there was emergence of scientific breeding principles, livestock, which shows and a kind of documentation of different types of animals and their characters and their characteristics. Okay, so and who brought genetics into limelight was actually Gregor Mendel in 1865. That was when he laid different foundation, a very solid foundation by his garden pea plant experiment. So he was the one that introduced what is called gene and heritability. We are going to see this as we go on. Then, what are the modern relevance of animal breeding? Number one, up to today, it is being projected an ambitious projection that is by the year 2050 there should be increase in animal protein by 70%. So that means up to today, animal breeding practice is still very relevant and is still going to be relevant even in the future. Then the modern relevance of today is as a result of the use of biotechnological tools such as genomic selection, marker assisted breeding principle, which is actually uh, which is actually to accelerate gains, that is to be able to improve in animal breeding. Then another one is sustainability agriculture. So the modern relevance up to today is breeding for feed efficiency, which reduces the use. And if you look at the screen here, we see that by the year 250, there's going to be increase in global food demand as well as other things. So that is one of the reasons why up to today, animal breeding is still very relevant in modern days. So application of this in Nigeria. Nigeria has been in the forefront where animal breeding practice is being done. And with the typical example of this is when you have West African dwarf, when you cross it with boa for faster, which has to do with faster growth in cattle, and also cattle improvement. That is the wild Fulani brand. They use it to they crossbred it with a Frisian in order to be able to enhance meat production. And we can also see this in a poultry program by introducing dual purpose breeds for meat as well as for egg. So it's not only going to be a a unit purpose is going to be for dual purpose, that is to be able to consume the meat of that poultry after. For example, layers can be used as meat as well as can also be reared for egg production. And a typical example of what we have in the Polytechnic Okeogun here has actually shown that 20% yield in well when you cross it with a goat. So in this polytechnic, consider that this is an example of application of animal breeding in Nigeria. And the objective of animal breeding are five, basically: higher fertility in terms of reproductivity, then better trade, that is some of these traits that can withstand environmental stress and environmental instability. Then it can also be used for baby production, that is the more babies you have, the more in terms of milk and meat production. Then heat adaptability, as well as it is also for economic purpose. Okay, production traits. So this is when the you come across different types of terminologies like growth rate, which is the speed at which an animal reaches market weight. Okay, the feed conversion ratio, that is FCR, that is the feed that is required per unit gain. And in terms of a milk production, that is the volume of milk produced per lactation period. Okay, then egg production, that is the number and quality of egg produced by layers per hen in terms of egg production. Functional traits. So functional traits, there are some traits that are functional, that is so for example, fertility is talking about conception rate as well as a calving interval. Then the next one is a mothering ability, which has to do with a care quality, offspring survival, and the rest. So in terms of longevity, that is productive lifespan of the animal, which has to do with that. Then calving and kidding interval, which is a time between successive births. Okay, that is when your animal give birth now, and in the next 9 months also give birth to another. So the time interval is actually known as a calving and kidding. So gene, which is we are very familiar with, which is the unit of heredity, which is composed of gene. So a gene is even a segment of DNA. So we have to know that. And whereas chromosome, which is the structural organizing gene within the cell nucleus, that is, you found the chromosome inside the nucleus. Okay, and when we come across a genotype versus phenotype, we know that genetic makeup versus, we are talking of genetic makeup versus the observable trait. And in Mendelian inheritance, so the first law of Mendel is actually known as a law of segregation, which has to do with allele pairs of separate allele pairs, that is, you during gamete. So just like you have it, that is, each trait is being controlled by a pair of allele or pair of gene. So the second law is known as the law of independent assortment, which has to do with gene, that is, different gene can assort, can independently behave of another. The fact that one is capital letter does not actually confer the fact that it must not be represented when it is going to be segregated. Okay, then the Punnett square at the Punnett square, this is an example of Punnett square. That is a tool which is actually used for predicting genotype ratio. Okay, and key genetic terminologies are allele. Allele is actually known as a referral form, that is the alternative form of any gene. So if you have a gene that is a tall, the alternative form of it is going to be shortness, and that is going to be called the allele. And when you come across dominant versus recessive, dominant is when you have the capital letter, which is going to be dominating. So in Mendel law and in animal breeding, there are some characters that are dominant, whereas the other one are recessive. And when we talk about homozygous, homo means say, zygous is from zygote. For example, capital A, that is a homozygote. And when you now have mixture, which is hetero, that is different zygote, that is a capital letter and small letter, called that. And the phenotypic ratio, that is the observable ratio versus the non observable ratio, that is the ratio that you can see with your naked eye, and that one that you cannot see, which is genetic in nature. And just like I said, trait pair, which has to do with Mendel actually performed his genetic principles on garden pea plants, because one of the reasons why he selected that was because garden pea plants, in terms of height, the height is being controlled by a gene. It is either it is tall or it is short. Then the seed shape, seed color, and etc. And the first filial generation after the crossing of the parents' gamete, the first filial generation are the one that will be produced and is going to be in heterozygous condition. Okay, and the second filial generation is being controlled, or you get second filial generation when you cross the product of the first filial generation together, and when you cross them together, genotypically it's going to be 1:1 ratio, and phenotypically it's going to be 3:1, just like this. So this is the parent, and okay. So example of a dominance is when you have in animal, that is a tall versus dwarf, so which is this one. So when you cross this one with this one, so what's going to be is that the first filial generation all are going to be tall phenotypically. And when you now cross the product of first filial generation together, you will now have um, 1:2:1. That is going to be this, this is going to be 2, and this is going to be 1. That is 1:2:1. Phenotype. And phenotypically, it's going to be tall to dwarf ratio is going to be 3:1. That is, 3 will be tall and 1 will only be uh, dwarf. Okay. Then application of animal breeding or genetics in animal breeding. You should know that in animal, there are certain conditions that are genetically controlled, and there are some that are not genetically controlled. For example, the coat color, which is a simple dominance in cattle, okay, and goats. So that is when you see goats, you see them in different colors. When you see cattle as well, you see them there in different colors. That means coat is an important tool or important trait that you can actually use in breeding them. Then the pole trait, absence of horn, or so when you see in cattle, you have some that naturally, some of them are hornless, that is polled. You don't call them unless you use polled. And those ones that are on. So when you now cross them together, it is also uh, very possible. So then you also come across what you call qualitative and quantitative traits. Qualitative traits are those ones that are controlled by very few genes or by one gene. For example, this coat color in goats is an example. And that of quantitative goods, those are the genetic traits that are being controlled by many, polygenic in nature, that is, that are controlled by many genes. And example of quantitative traits is also continuous generation, such as continuous variation, such as weight as well. So, characteristics of qualitative traits are Mendelian in nature. That is, they obey Mendel principles. Then, environment has a very small, reduced influence on qualitative traits. An example of that is the coat color and presence or absence of horn. And characteristics of quantitative traits, these are traits that are being controlled by many genes, just like I said, and environmental play very significant role on their performance. An example of that is nutrition as well as nutrition management. So example of this is a milk yield, growth rate, and litter size. And as you can see it, there is a kind of gradual improvement on the production. That is telling you that it is being controlled by many genes. Okay, an example of this trait in livestock, that is quantitative, is in terms of milk, which is measured in liters per lactation. Then the on status, for example, this one is a qualitative, whether it is a present or it is absent. Then growth rate, this one growth rate is quantitative because it is measured in kg per day. That is, when you start weighing them every day. So the average weight gain is what is going to be the growth rate. Heritability. Heritability actually means the proportion of phenotypic variance, which is as a result of a genetic variance, and it actually ranges from 0 to 1 in all genetic possibility. Okay, high heritability traits respond quickly to selection, and just like what we have in Nigeria, that is, we have both show heritable heritability. Okay, which Nigerian draft goats show heritability of 0.35 in terms of milk production. So then example of heritability, that is a milk yield in cattle, which has these scores. So it is better we document it that the the higher the number between 0 and 1, the higher it is tending towards 1, the more heritable it is. So that means anyone with a 0.25 is of lower heritable score than the one that is very close to 1, like 0.75 and so on. Okay. So genetic variation, what can cause genetic variation? Mutation, recombination, and migration. One of the powerful tools of animal breeding is mutation. If there is no mutation, there cannot be variability, there cannot be selectivity, there cannot be heritability, and there cannot be uh, breeding as one will actually determine. So mutation is very powerful. That is sudden change in genetic makeup, and genetic recombination is also very possible. That is, you get the gene, good gene from one of the animal, then you crossbreed it with another one. When two of them will now meet together during meiotic division, and that is actually regarded as a recombination. And heritability or genetic variation also occurs when animal migrate from one place to another. So, which means for us to be able to maintain, for us to be able to continue in animal breeding, that means we should be able to be allowing variability. We should be able to allow animals, all these animals, to move from one place to another. So as to uh, give us the selected trait that we are actually looking for. So maintaining genetic diversity. So rotational crossbreeding system is another very powerful tool that people use. That is a system that involves mating females of one breed to male of another in a planned sequence. So you can see now, this is breed A. It is meant to interact first with B, then to produce breed B. So when you now rotate this over successful period of time, it is known as you are trying to maintain genetic diversity. So over time, there's going to be constant. Then the use of a conservation for flocks. So that is when you see some uniqueness. For example, when you crossbreed your animal in the successful generation, if you now have already improved yield animal or improve yielded animal, how will you maintain it? How will you conserve it? And that is when genetic conservation of flocks will actually come in. And that is um, you specifically manage the population to preserve unique genetic resources. Okay, and cryopreservation of gamete and embryo, just like you see here, this is a sperm from male animal, and this is the oocyte from female animal. So when you now cross, you mix them together, you get embryo, and this embryo you can preserve them in frozen condition for over a long period of time. So in this lecture, we have been able to learn some basic concepts and basic definition. We also learn how to use Mendelian rules in animal breeding. What are these Mendelian rules? What are the traits that are very, very possible? What do we mean by heritability, qualitative versus quantitative? So that is exactly what we have done. In the next lecture, we are going to do, we are going to continue with the selection method. Selection method. And if you want to read more on this, that is, we'll go to